Hello friends, today we'll be looking at the diverse and crucial process of protein synthesis, the process through which proteins are synthesized in our cells. Protein synthesis is one of the most important metabolic processes in all living organisms. The central dogma of protein synthesis, from DNA to RNA to protein, is mostly conserved amongst all species. There are many complex cellular structures and sub-processes that must work synchronously for protein synthesis to occur properly. In this video, we will talk about the fundamental processes of protein synthesis, notably transcription and translation. This will be followed by post-translational modifications, finally concluding with protein distribution and trafficking. So we will start off by describing the fundamental processes of protein synthesis, which is composed of two main parts, first of which is transcription. Transcription is the process by which coding DNA from a gene is transcribed into a complementary messenger RNA, also known as mRNA. This takes place within the nucleus. Spliceosomes then cut out introns from the mRNA and piece together the exons. A poly A tail and 5' cap are also added to the mRNA. The final mRNA is then transported to the cytoplasm, where the process of translation can occur. Transcription is followed by the second part of the process of protein synthesis, translation. Translation is the stage in protein synthesis where the mRNA joins with ribosome subunits and the first transfer RNA, tRNA, with its corresponding amino acid, the matching nucleotide sequence on the mRNA transcribed from the DNA. This is the process of initiation. Both prokaryotic and eukaryotic chromosomes contain a small and a large subunit, but these are of different sizes depending on the type of organism. Eukaryotes use an 80S ribosome with a 40S and 60S subunits, while prokaryotes use a 70S ribosome with 30S and 50S subunits. The tRNA for formal methionine or methionine has the anticodon UAC and pairs with a small subunit. Then after this union, the large subunit joins with the tRNA and small subunit to form the full ribosome and finish initiation. Elongation describes when tRNAs are fed through the ribosome and their amino acids form peptide bonds. There are three sites: the amino acetyl site or the A site, the peptidyl site, the P site, and the exit site. The F-met tRNA starts in the P site, and the next tRNA enters the A site. From there, the enzyme peptidyl transferase is used to form the peptide bond between the two amino acids. After this, the tRNA in the P site goes to the E site and leaves the ribosome. The A site tRNA goes to the P site, and a new tRNA enters the A site. This process continues until the end of the polypeptide, where termination occurs. Termination is when ribosome subunits detach and the polypeptide is released by a releasing factor, when the N nucleotide combination is signaled on the RNA strand. Now we will discuss about the post-translational modifications. There are many post-translational modifications that can take place. Many of these modifications occur in the Golgi apparatus and/or in the rough endoplasmic reticulum and involve the addition of specific groups to the protein. Some major groups that can be added to a protein include carbohydrates through the process of glycosylation, lipids through the process of prenylation, and covalently bound metal-containing groups, or prosthetic groups. In addition to these additions, proteins can also be cleaved into smaller units to make them functionally active. For example, insulin is synthesized as pre-proinsulin, which is then cleaved into proinsulin and finally processed into insulin for its functional use. And finally, we will expand on protein distribution and trafficking. Proteins can be distributed to one of three major locations: the cytoplasm, the nucleus, or the external environment. Each of these destinations has a specific protein trafficking pathway associated with them, mediated by the vast endomembrane system. Proteins to be used in the cytoplasm or nucleus are synthesized on cytoplasmic ribosomes and simply released into the cytoplasm upon completion of translation. From here, the nuclear proteins can enter the nucleus through nuclear pore complexes. The proteins destined for export outside the cell or insertion into the membrane are synthesized on ribosomes bound to the rough ER and are then channeled through the Golgi apparatus and into vacuoles for exocytosis, the process by which the product is transported through the plasma membrane to the extracellular space.